Recording in progress.
Can everyone see this okay? All right,
Yes.
Thanks for coming, everybody. I'm going to talk to you about my dissertation, Thought for Food, that is brain activity and task performance of American crows in pursuit of a food reward. So I guess in order to get started, hang on a second, let me fix this. All right, so in order to get started, I should probably tell you about why it's important to study bird brains. And a main reason for that is due to one Ludwig Edinger, who was a comparative anatomist from the late 1800s to early 1900s. Now, Edinger was a very influential person. He actually started the field of neuroscience, or at least he was one of the main figures in that comparative neuropsychology or neuroscience field. However, as part of his job, he compared a lot of different brains from different taxa. And whenever he got to birds, he erroneously concluded that the majority of the bird brain was composed of striatum, which is the part of the brain that's associated with instinctual behaviors or species-specific behaviors. Now, in actuality, that part of the brain is the pallium, which is what's used for higher order cognitive thought. So he thought the birds were simpletons, and that reputation persisted among the scientific and general public community for quite some time, for several decades. That's where the term bird brain came from. However, birds are actually quite intelligent. We know that they are capable of a lot of types of advanced intelligence comparable to mammals. And so you can see a list here on screen, but just as one example, this bird right here, his name is Alex, he's an African gray parrot. His trainer taught him to speak different human words. And Alex demonstrated that he could categorize the world based off of different features. So for example, you could present Alex with this tray of objects and ask him, how many blue? And he would respond 2. How many orange? He would respond 4. He could categorize things based off of their color, their materials, what shape they were. And so that really shows that he could categorize the world around him. And that's just one of many different things that birds are capable of doing. Now, one of the other reasons why Edinger erroneously messed up was because the bird brains are structurally very different from mammalian brains. And that's a result of more than 300 million years of evolution since the last common ancestor shared by both taxa. And as a result, bird brains are capable of things that mammalian brains are not capable of. So for example, birds can sleep half of their hemisphere while the other, or one half of their brain, one hemisphere, while the other half remains awake to scan for predators. Mammals can't do that. Birds can also generate new neurons throughout their life. In fact, some birds even have seasonal plasticity, where parts of their brain grow or shrink based off of the need of the moment. Caching birds, for example, might increase their memory whenever they have to find caches of food to survive the winter, but then that same part of the brain shrinks in the spring when they don't need it anymore. Humans can't do that. However, despite all these structural differences and these unique capabilities, functionally, bird and mammalian brains are very similar. And so it's important to study different types of brains to see how they do the same sorts of features. It's sort of like comparing a Mac to a PC. Both of them have different operating systems, but they do effectively the same thing on the user end. And so we can learn more about ourselves by learning more about how bird brains process the world. So I was interested in studying this, and that makes American crows a model species for this type of research. Crows are intelligent, so you can give them novel tasks and see how they solve it. They're social, so you can look at the effect of different social factors have on their ability to do things. They know how to solve novel tasks. They even practice a proto-tool behavior. They'll drop objects on the ground in order to crack them, things like hard-shelled nuts, or snails or clams. They'll drop them onto a hard surface in order to break them open. It's not quite tool use, but it's on the way there. Another advantage that crows have is that they're local. So I don't have to go outside of the state to find these animals. They're all over Seattle. And one other advantage that they have is that there's a good foundational amount of prior literature. We know a good deal about how crow brains act with PET imaging. We know a good deal about how crow social behavior works, but we don't know everything. There's still so much that we don't know about crows, which gives me an opportunity to find out more. For example, we know that they use social learning to learn about sources of danger, but can they use social learning to learn about how to acquire new sources of food? What's the extent of their ability to use tools? And what sorts of characteristics are shared by successful individuals? And I'll define successful individuals for each of the studies that I actually examined this in. Successful is a subjective term. So the goals of the studies that make up my PhD, the overall goals anyway, are to examine how these birds and their brains process cognitively difficult challenges. And slightly more specifically to crows, I want to identify the different characteristics that are shared by these successful individual birds. And in order to accomplish these three goals, I did 3 separate studies. In the first study, I examined the different social and individual characteristics of birds that were best able to solve a string pull task, a novel task. For the second study, I looked at the brain activity of crows as they listened to conspecific vocalizing under different circumstances, with and without the presence of food, and also just what's their brain doing when it sees food without conspecific vocalizations. And for the final study, I combined elements from both of the previous studies and looked at the physiological and neurological factors that are associated with tool use, and which of these come into play as these birds learn to use tools to solve a novel task. So let's get into this. For the first study, I have already published this in a special edition of Ethology. It's titled Individual and Social Factors Affecting the Ability of American Crows to Solve and Master a String Pulling Task. Now, with not just bird research, but with any sort of research that examines an animal's intelligence, a very common tactic to see how intelligent the animal is, how it understands the world, and just what the limits of its intelligence are, is to give it a novel task and see how it solves it. How long does it take? What strategies does it use? Can it solve it at all? And for this study, I gave the birds what's known as a string pull task. Now for this task, you have a piece of food that's hung from a perch with a piece of string. And the goal of the bird is to retrieve that food. And they utilize different tactics to get this food. So one method is what I call the food intercept method. That's where the bird flies to the food and intercepts it from midair. Now, this method is a bit difficult. It requires a good deal of agility, and so it's more difficult for larger birds to do. And additionally, if the bird doesn't retrieve the entire bit of food, then the remainder will swing back and forth like a pendulum from the force of pulling a piece of it away. And so the bird has to wait for it to stop before it can try to retrieve more. Another method is called the attacking string method. That's where the bird goes to the perch where the food is attached to the string, and then it begins attacking it. It pecks it, it hammers it, it pulls on it, and eventually the string breaks, it falls to the floor, and the crow is able to fly down and retrieve its prize. This method requires a good deal of physical strength, and so it's difficult for smaller birds to do, and it takes some time. The final method that the birds could use is called the string pull method. And that's where the bird goes to where the food is attached to the perch, and it reaches down, grabs a string, and pulls it up. If it lets go of it, though, it'll fall back. So it steps on the string to hold it and hold the slack, and then it reaches down, pulls up more, steps on it, and through this repetitive action, it ratchets the food up to where it can easily get it. It's cognitively difficult. Most birds don't know how to do this at first, but once they learn it, it's easy and it's quick, and it doesn't discriminate or it doesn't bias bigger or smaller birds. Here's an example. So the camera's on the ground pointing upwards, and you can see this bird, I believe this one's dark blue, and it reaches down, pulls it up, and steps on it. And it repeats that action. Now notice that its neighbor is paying attention to what it's doing. And that's important because crows are social animals. And we know that they can spread knowledge about dangerous predators through social learning. But can they learn how to solve novel tasks by watching their conspecifics do it? So that's what I was curious about. I was also curious, what are the individual factors of what are the physical factors of the individuals that are most successful at this task? Certain features about an individual might give it an advantage. So for example, adults who have lived longer might have had more opportunities to experience something or encounter a challenge and overcome it that might give it an advantage in this task over younger individuals who are more naive to the world. The animal's body condition, as another example, might be important. If it's starving, for example, it will be much more motivated to do whatever it takes to get that food. And so it'll interact with the task for much longer than a well-fed individual would. And as one final example, perhaps the brain size might be important. Some of the studies that I have listed here have found that individuals with larger brains are better able to solve novel tasks than individuals with smaller brains. So I included that as a thing to look into. So here's how I did this. I captured 27 crows over a 3-year period, and I housed them in an enclosed outdoor aviary. And I divided them into 2 groups. The birds that were put into the model group were put into even-numbered cages, and they were given access to the task from day 1. As soon as they were brought into captivity, they had access to it. The other birds, the other half, were put into the observer category. They were put into the odd-numbered cages, so we had observer, model. They were interspaced like that. And they had to wait until their neighbor fully mastered the task. The idea being that they could watch as their neighbors learned to solve it, and if they could learn to solve it themselves using social learning, they would solve it more rapidly, and they'd be more likely to solve it when they first had access to it. I also examined 7 individual factors. I thought that adults would be better for the reasons I described otherwise, and I used feather and mouth color to sort birds into adults, which are older than 3 years, and subadults, which are in their 2nd year of life. Primarily, subadults have pink in the inside of their mouths, and they've got brown and dull black feathers, whereas adults have all black mouths and sleek, glossy black feathers. I used Pullman length as a measure of size. That's just the measure from the base to the tip of their beak, or their Pullman. I measured their level of nervousness by standing in front of their cage for 1 minute and measuring how often the bird moved back and forth. My rationale being that calmer birds would be better able to solve this task because they are less stressed overall. I measured sex by taking a blood sample and then having one of my technicians run a PCR or run gel electrophoresis on the PCR product. Basically, females have ZW chromosomes and males have ZZ chromosomes, and gel electrophoresis can detect that. I looked at body condition, and to calculate that, I ran a linear model on the regression of Coleman length against the body weight and took the residuals. So if you have a smaller individual, but it has a higher weight than expected, it would have a positive value for body condition. And finally, I looked at absolute and relative brain volume. And I calculated that by taking the brain volume of CT scans that I'd obtained with these birds as part of a later study, which I'll describe later on in this presentation. And then for the relative brain volume, I just, again, took the residuals of the Pullman length, so body size, against absolute brain volume. I ran a model selection on these values using GLM models and AICC. I considered any model with a AICC value of within 2 of the best model to be competitive. And so I compared the 1 social factor, the 7 individual factors, and a null model against their probability of initially solving it when they first gained access to it, and the time it took them to fully master this task. And finally, I came up with a difference index. This calculation, or this equation, calculates and quantifies how similar or different the method choices are of 2 individual birds, or 2 adjacent birds. So you have the percentage that an observer and the percentage of a model and their proportion of successful trials for food intercept, attacking string, and string pull methods. This equation returns a value between 0 and 1, where 0 means these 2 birds are completely similar. They use the exact same methods overall. And 1 means that these 2 birds used completely different methods. They are completely dissimilar. So let me show you what I found. So for the model selection, I found that none of my models strongly explained the weight of evidence, and none of them were better than the null model. But I did observe this weak trend of social role being almost the best model. In fact, it was the best model, but it was still competitive with the null model. However, it was reversed from what I thought would happen. The observers were worse at this task than the models were. In terms of time to master, I found 4 factors that mattered. Social role, again, the observers were worse than the models. They required a variable amount of time, whereas the models consistently solved in a short amount of time. Absolute value or absolute brain volume and relative brain volume were also important. Smaller brained birds required a variable amount of time, whereas larger brained birds took a consistently small amount of time. And finally, body condition was also important. Birds with low body condition had more variable amounts of time required to solve, whereas birds with high body condition solved consistently in a short amount of time. And the last major finding that I found in this study is that the observers were more likely to copy the model's method choice. So what's all this mean? Well, let's start with the most important factor, and that is that these crows are capable of limited social learning to solve this task. They weren't better at it compared to the birds that didn't have other birds to model it. But once they learned it, they were able to refine their technique by using the techniques that the models were showing them. But that does beg the question, why are the observers worse at this than the models were? And I believe this is because the observers were less hungry than the models. They'd been in captivity for 2 weeks, whereas the models were given this task as soon as they were captured. They were brought in from the wild. They were still hungry. They had poor body condition. Whereas the observers, they'd spent 2 weeks being fed. They learned that they were going to be fed every single day. And so they were just less motivated to do this task, to retrieve food when they were already being fed. And finally, I believe that the more successful birds, the models, had larger brain volumes because they were predominantly male. So by pure chance, I happened to assign more males to the model group than the observer group. And that was random. I didn't know what their sex was until after I'd released them and the study was concluded. So male crows are bigger than female crows. And there's a positive correlation between body size and brain size. And so the bigger male birds had correspondingly bigger brains. So that's why I think that there's that effect of brain size against ability to solve the task. All right, so let's talk about brain activity and how they respond to vocalizations and food. So as I mentioned earlier, crows are social animals, and social behavior is cognitively complex for the brain to process. To use American crows as an example, we have communal feeding events where there'll be a piece of food or a large pile of food that's just out, and the crows will find it and they'll gather in large numbers. And at these events, there's a lot going on. Each individual bird is trying to secure some access to the food for themselves. There might be fights between large dominant individuals for access to the food. You have birds assessing one another to determine whether or not they are dominant or subordinate to the other. You have birds that are trying to court others. They're trying to learn more about their neighbors. They might be trying to recruit allies to the food so that they can share. There's a lot going on. And during this entire time, they have to be on the lookout for predators. That's cognitively complex stuff. Additionally, they're receiving a flood of visual and auditory information. They're communicating, they're vocalizing. They're also posturing and giving visual displays. How does their brain handle all this information? I can't just look at one individual part of the brain because there's interactions between different regions. So I had to use a holistic approach to examine this. That's where positron emission tomography, or PET imaging, comes into play. It's very useful for this type of research. The way it works is that you inject the subject with a radio tracer, and that radio tracer is absorbed into the body metabolically, and it emits positrons, which the imager can detect. Now, brains require a good deal of energy. They're hungry for glucose. And so if you use a radio tracer like 18-FDG, which the body treats as glucose, I can use it as a proxy for brain activity. You inject the bird, its brain starts working. Whichever parts of the brain are working harder will be consuming more energy, absorbing relatively more glucose, and it becomes fixed. And then the positrons are emitted, and the imager can detect them. So for this study, I captured crows and I mimicked a feeding event, and I looked at their brain activity against a baseline control. So for the baseline scan, I just put them in an empty box. There's no added sound, no additional visual stimulus. And then I added, for one stimulus condition, I added food to see how they responded to that. For the audio condition, I added the sound of crows vocalizing at a food source. And finally, to mimic a true feeding event, I played the sound of crows vocalizing while the crow was simultaneously looking at a food source. I hypothesized that for the visual stimulus, the hypothalamus would be active. That's a region of the brain that's involved with motivation and food regulation. For the audio stimulus, I believe that the nucleus tenea of the amygdala, or TNA, and the lateral septum would be active. Those regions are associated with social interaction. If the calls were encoding information about food, then I also thought that I'd see some activity in the hypothalamus, again, because it's involved with food regulation. And finally, for the combined stimulus, I believe that I'd see activity in all of the above regions, and also potentially the thalamus. The thalamus filters and organizes and relays information from the senses to different parts of the brain that know what to do with it. And so I figured with the additional sensory information, we'd see additional activity in the thalamus. I used 13 crows for this experiment. These are actually some of the same birds that we used for the string pull task. And well, I recorded the audio of birds vocalizing at the capture site one day before I caught them. So I've got a good sample of the audio that crows give when they're vocalizing and when they're eating at a food site. And while they're in captivity, I determined what their favorite food was. It's fried chicken. They love that stuff. I covered it in a plastic wrap so that I would minimize any confound of scent. So they got used to receiving their food like that. So on the day of the experiment, I took these birds, I injected them with a radio tracer, and while they were metabolizing that radio tracer, I exposed the stimulus to them over the course of 10 minutes. And I did so by alternatingly revealing the stimulus for 60 seconds and then hiding it for 30 seconds. This reveal and hide of the stimulus kept the bird interested. It gave it multiple opportunities to calm down and reset and then re-expose itself to the stimulus. So the brain didn't get bored. It had multiple opportunities for each pertinent region to absorb more of that FDG. Then we anesthetized the crow, so it was sleeping and immobile. We strapped it to the multimodality bed, and we ran the PET at 26 minutes after injection. To analyze the data that I collected from PET, I used a program called Neurostat. And this program takes a global normalized or compares the difference in FDG detection after it's normalized it to global values. It effectively does a z-test using the pooled variance of all the birds in the study, and it's comparing each individual voxel. Now there's thousands of voxels, so it applies a modified Bonferroni correction to the p-value with some smoothing factor. It compares the adjacent voxels with it when it makes that comparison. So it's not comparing a voxel here against a voxel way over here. It's more so comparing this voxel against the surrounding voxels. And I further verified these results by sampling volumes of interest at the coordinates of peak activity, just to make sure that this wasn't being driven by one outlier individual. Here's what I found. Whenever the crows saw the visual stimulus of just the food, there was hardly any change in brain activity. But when they heard the audio of crows vocalizing, that did cause a change in brain activity. I saw increased activity along the border between the nidopallium, the medial nidopallium, and the lateral septum. I saw activity in the medial striatum, and I saw activity towards the back of the brain in the caudal nidopallium, sort of adjacent to and possibly even including field L, which is a region that's associated with audio processing. So the lateral septum is involved with social interaction, especially aggression. And the medial nidopallium and the field L are both involved with audio processing. And the medial striatum is also involved with audio processing, but all the studies that I found looked at a different part of the brain than the activity that I observed. So I'm not quite sure what to make of that. And then most interesting, the combination of seeing the food and hearing the vocalization caused some emergent brain activity. I saw activity in areas that weren't active for either of the individual stimulus by themselves. I saw activity in the nucleus tenea of the amygdala, or TNA. I saw activity in the medial nidopallium, the lateral septum, which I did observe in the previous condition. And I also saw activity in a different region of the caudal nidopallium. That's more medial to the previous condition. Now, the TNA and the lateral septum are both involved with social information. The medial nidopallium and the caudomedial nidopallium, that's the area of the back of the brain that I observed. Those are both involved with audio processing. So why did I see extra activity whenever I introduced the food to the sounds that I didn't see when I saw the food by itself? Well, to me, that says that the TNA and the caudomedial nidopallium, those are both of the emergent activity regions, they must be involved with either multimodal processing or with integrating sensory information with social information. And based off the literature, I believe that the TNA is involved with the integration of social and sensory information, and the caudomedial nidopallium is involved with multimodal processing. So it's got auditory and visual information, and it's got to combine them. Now, I was also curious, why didn't I see any activity for the food by itself? I mean, the crow needs food, it's essential for survival, so why didn't the brain care about it? I believe that, or at least the evidence suggests that the crow considered the food to be less interesting than the audio stimulus. The food didn't add any additional light, it remained static, and it was something that the crows were used to receiving in the context of being in the imaging lab. They were fed after they received their baseline scan, and they'd been receiving food over the course of their captivity. So that wasn't highly unusual for them. Whereas the sound, I was adding additional sounds to their environment. So the volume was increased, so to speak. The sounds were dynamic. They were vocalizing, they were calling. There are different types of vocalizations in there. And I imagine for the bird, it was very odd to hear their flockmates vocalize, and they were hearing outside while they were stuck indoors in a cage. That was probably surprising to them compared to the food. Additionally, the brain had to work harder for the sound or for the vocalizations compared to the food. The food provided its information directly. The food was food. The crow just saw it and it was there. Whereas the vocalizations, if the vocalizations were encoding information about food, there's one extra step of processing required. The vocalizations were information about food. They weren't the food itself. And finally, there might have been all sorts of social information embedded, and I'm pretty sure there were lots of social information embedded in those vocalizations. The food, well, the food was food. The crow saw it, we knew what the crow was thinking about. Whereas the vocalizations, well, they might be encoding information about courtship, about agonistic encounters, about fights and other individual information, and we just don't know. The activity of the lateral septum, because it's involved with regulating aggressive behavior, does imply that there was some aggressive interaction and vocalizations were given and the crow could detect it. So a lot of useful information there. And finally, the third chapter of my dissertation, I combined elements of both of my previous chapters, and I looked at the individual characteristics of successful birds and their neurological activity as they learn to solve a task requiring the use of tools. Now, tools, let me briefly define that. Tool use behavior is where the subject manipulates some environmental object in order to alter the condition of another object for their benefit. So to use chimpanzees, for example, they will use sticks to fish for termites. They're using an environmental object to manipulate it, or they're manipulating an environmental object to alter the condition of another object. They stick it in there, termites grab it, and now they have access to the termites. It benefits them. Relatively few species use tools, but the species that do are distributed over a wide variety of different taxa. Cetaceans like dolphins use tools. Primates use tools. Birds use tools. So the best known example are New Caledonian crows, which is a photo available on my slide. These birds use two different types of tools. They use pandanus leaves to fish out grubs from small little crevasses that they can't access easily. And they also use spines to spear them and pull them out. They just innately do it. And in fact, some researchers looked at their brain. They've found enlarged areas in their brains compared to other crows or other corvids that don't use tools. Now that actually makes American crows ideal for this type of study because they rarely use tools in the wild, but they do practice behavior that is almost tool use. We call that proto-tool behavior. And in the case of American crows, they'll grab an object like a clam or a snail, and they'll drop it from a height. It hits the ground and breaks, and they can gain access to food there. That's not quite tool use behavior. Tool use behavior would be if they took a rock and they banged on it with a rock. But they can learn to use tools in captivity. So I was very curious, how do they transition from this proto-tool behavior into true tool use behavior? And I was curious about what's going on in their brain as they make that transition. So I decided to use a tale from antiquity for this. This type of task is called the Aesop task, and it's based off of the old Aesop fable, The Crow and the Pitcher. Now, according to this story, a thirsty crow finds a pitcher that has water in it, but the water is low. It can't reach it to quench its thirst. So it picks up stones off the ground, drops the stones into the vase. The stones displace the water upwards until eventually the crow can reach the water and quench its thirst. That's considered tool use. The crow is using stones to alter the condition of another object to their benefit. Now, this type of task has been given to multiple corvid species. They've found that rooks, ravens, New Caledonian crows can solve this task. And so that told me that American crows could most likely learn to solve this. So here's what I thought would happen. I hypothesized that I'd see increased activity in the mesopallium, the tegmentum, and the striatal complex. Those are regions that were enlarged in the New Caledonian crow, which is a bird that uses tools throughout its landscape. I also thought I'd see activity in the NCL. That's a region in the very back of the brain that's been compared to the prefrontal cortex of mammals. It's the area where higher order thinking happens and the crow makes executive decisions. And I also thought I'd see some activity in the hippocampus. That's a part of the brain that's associated with memory. I thought this because the crows learned to solve this task, so they'd probably be remembering their past experiences whenever they saw this in the imaging lab. So I utilized a lot of the same factors that I did in the previous experiments. I looked at those same individual physical characteristics. I already explained how they might be applicable to crow behavior and how I measured them. So in the interest of time, I'll skip that. And I also looked at their brains using PET imaging, again, using the same methodology that I previously described. So here's how I did this experiment. I captured crows in the wild and brought them into an outdoor aviary. And for the 2 weeks or so that they were in that aviary, I habituated them to the conditions that they would experience in the imaging lab. I would catch them, I'd put them into a small cage, and I'd put a white training box in front of them. They couldn't leave the cage, but they became habituated to being in that small space and looking into a white box where there could be something in it. I then brought them to the imaging lab and I scanned their brains using PET imaging. And this time, for this scan, that was the first time that they were exposed to this ASOP task. And I'll explain more about that in just a moment. So they didn't know what it was. They were naive to it. And I used that as my baseline scan. Then I put them back in the aviary, and for the next several weeks or months, I trained them to solve this task to varying degrees of proficiency. And I'll explain exactly how I trained them in just a moment. After they learned to solve the task to varying degrees of success, I brought them back to the imaging lab where I showed them the same exact stimulus, the ASOP tube, but unlike their first scan where they didn't know what it was, this time around they were knowledgeable about it. Then I brought them back to the aviary, and a few days later, I let them go. I used 16 crows for this study, and I made multiple comparisons between different groups. I compared the naive crows overall against the knowledgeable crows overall, but then I also subdivided them based off of their proficiency. So some birds became very good at this task, some birds were less good at it. And so I sorted them into an ordinal proficiency scale based off of their proficiency, and I compared between those groups. So I trained the birds using a series of incremental steps. First, or after they had their first scan, I began training them. I got them used to leaving their small little training cage, entering the arena, and eating food. Once they did that, I introduced an ASOP tube to their environment. I made it very easy to solve. The water is all the way up at the very top. The crows would just take the food out, very easy. The whole point of this is just to get them used to the idea that they could get food from the ASOP tube and to overcome their neophobia of it. Then I began lowering the water level. I increased the depth. And I determined just what depth was the depth at which the crow could no longer reach the food. No matter how much they reached, they couldn't get it. Any bird that was stuck at this stage and could not progress any further was considered to be in the low proficiency category. The birds that were able to progress, I introduced training stones. Now these stones were attached to the interior of the tube using fishing lines. So if the crow bumped them, they would fall in, they could not fall outwards. And so the crows would inadvertently bump into them as they tried to reach the food. The stones would fall in, the water level would rise, and the crow would be able to see it and hopefully make the connection that stones go in equals good. So any birds that learn to drop the balance training stones, I progress them to the next step. Or I'm sorry, any bird that learned to balance the training stones or any bird that learned to drop the balance training stones, I consider to be medium proficiency. Once they did drop the balance training stones, I increased the length of the fishing lines. These stones are now hanging on the exterior of the tube. If the crow bumps into them, they'll just be moved out of the way. They won't fall into the tube. The crow has to learn that they have to pick up the stones and drop them in. Any crows that do that, well, it's a very quick and easy progression to learn to, okay, the stones are on the outside, they need to go in order for me to get the food. And from there, they quickly learn, hey, all these stones that have been around this entire time, I can drop them into the tube. Once they've done that, I consider them to have fully mastered this task, and I put them in the high proficiency group. And whoops, yes, I measure their individual factors and I image them the same way as I did in chapter 1 and chapter 2. Model selection, I use multinomial and binomial models, and I use whole brain voxel-wise subtraction and VOI verification for the brain stuff. Now, 4 birds could not progress beyond low proficiency. 8 of the birds got to the point where they were accidentally dropping stones into the tube, but they weren't deliberately dropping the hanging stones. But 4 birds fully mastered the task and could solve it without any training stones. Here's an example. So the bird drops the stone, but it still can't quite reach. Needs another one. The angle's a little awkward, but the second attempt drops it in, and this time it's able to retrieve the food item. Now, due to some technical issues, I wasn't quite able to get all of these birds imaged. We had some issues with the scan, so my sample size for the model selection was all 16 birds, but for the PET imaging, I could only use 14 birds. I had 3 birds for the low proficiency and 7 birds for the medium proficiency. So here's what I found. The individual factors that most mattered and most applied or most best predicted the ability of these crows to solve this task is age. All of the birds that were in the high proficiency group were adults. None of the sub-adults were fully able to master this task. And so because the adults were the only birds that were able to solve it, I reran this model selection by excluding the sub-adults and just looked at what factors best predict their ability to solve, binomial. And within the adults, I found that the successful adults that were in the high proficiency group, they had smaller brains, smaller bodies, and were mostly female. But the mostly female part was the most important, because every single adult female I had in my sample solved this task, whereas only 20% of the adult males learned to solve it fully. And I mentioned this earlier, but sex in crows is strongly correlated with body size and brain volume. So these birds weren't female because they were small because they were female. So the fact that they were female was the most important part of that finding. So why is that? Why were adults the most successful at solving this task? I believe that because crows can live for so long, they've had many more opportunities to encounter challenges in their life, and those challenges primed them to be better able to solve this task. Additionally, females compared to males, I mentioned this before, but they're smaller, which makes them subordinate when it comes to gaining access to food. And so they've had to rely more so on their brain to gain access to food, whereas the larger males can just rely on their status. They're bigger, they walk up, they force their way into the food pile and they get their food. Whereas the female, well, it has lots of strategies it can use. It can wait until the males are distracted before it jumps in and grabs something. It can try to steal food from a bird that's grabbed something and moved to the periphery. It might try to cause an opportunity by recruiting allies. It might even mentally calculate, okay, I'm not going to get in here for a while. It might be better for me to just leave and find food somewhere else. And additionally, this finding is consistent with other animals. Dolphins use tools, and it's predominantly used by females. The same with chimpanzees and bonobos. And both of those animals, the females are subordinate to the males. Now let's talk about the PET imaging results. Now, my committee might be familiar with this, but I spent months trying to deal with some technical difficulties with these scans. These brains just were not aligning properly. We kept getting these artifacts along the edge because they were being stretched too much. They were aligned improperly. And come to find out, that was because their brains were shrinking. So during the post-training scan, their brains were on average 5% smaller than they were during the pre-training scan. So I'm going to look into that in a future study, but for now, we solved this problem by treating the brains as independent rather than paired, even though they came from the same individual. That solved the technical difficulties, even though it lowered our ability to statistically detect changes. So let's talk about what I found. So just looking at all of the crows, I'm not differentiating them based off of their proficiency. When they were first exposed to the ASOP tube, they had a lot of activity that wasn't there the second time around. I saw lots of activity all throughout the brain, in the mesopallium, in the nidopallium, and all the way into the back of the brain in the nidopallium caudale. Those areas are associated with executive planning and higher order cognition. So evidently, these birds, their brains were in overdrive when they were naive and they were first exposed to it. But then during their second scan, after months of exposure to it, there was no significant difference there. So I looked into what about differences based off of proficiency? So within the second scan, the post-training scan, I saw that the medium proficiency birds had increased activity in their mesopallium compared to the high proficiency birds. And the low proficiency birds had increased activity in their hypopallium apicale and in their mesopallium ventrolaterales, or MVL, compared to the high proficiency birds. The mesopallium is associated with higher order cognitive thought, just like with the pre-scan versus the pre-training scan versus the post-training scan. Whereas the MVL and the HA, the hypopallium apicale, those are associated with visual processing. So evidently, the medium proficiency birds are still thinking about this task quite a bit, at least compared to the high proficiency birds. Whereas the low proficiency birds, they weren't doing a whole lot of higher order thinking, they were just staring at it. Let's talk about the high proficiency birds. Those females, those adult females, their brains were active, but different compared to the others. So compared to the low proficiency birds, they had a lot of activity in the hippocampus, in the nucleus basal-rostralis, and in the cerebellum. And compared to their past selves, compared to their pre-training scans, they had a lot of activity in their tegmentum. Now the cerebellum and the hippocampus, primarily the hippocampus, but also the cerebellum, those are involved with memory. Additionally, the cerebellum and the tegmentum, those are involved with motor coordination. And finally, the nucleus basal-rostralis, that's associated with processing sensation from the beat. So, what to make of all this? Well, the naive and the less proficient birds, they use the higher order cognitive processing centers of their brain. Evidently, they're trying to understand what that tube is, how to retrieve the food. For the naive birds, we might even see some activity because of neophobia. Crows are afraid of things that they're not familiar with. And so they could be trying to figure out, is this thing, which I don't know what it is it a threat to me? But even so, even the birds that are familiar with it, we still see activity in the mesopallium compared to the birds that are better at it. Those birds that were better at it, well, instead of using the higher order cognitive thought processing centers, they switch over to muscle memory. There's no longer any need for those high power parts of the brain because they already know how to solve it. In fact, their brain activity, the shift from executive function into muscle memory and motor control, that strongly resembles the same shift in brain activity that you see in humans as humans learn a skill. So some research has looked into human athletes, and novices use a lot of the prefrontal cortex, which is associated with executive planning and higher order thought processes, whereas the experts, the elite athletes, they rely more on muscle memory. So again, structural differences, but comparable function. This suggests that with birds and possibly other animals, they need those higher order circuitry or circuits to learn the task. But once they've learned it, then all they really need is just motor control and tactile feedback to perfect their technique. If an animal is helped to learn a task, once it's learned it, it can practice it on its own. And these findings are consistent with the research that found that New Caledonian crows, those crows that use tools all the time, they have enlarged brain areas that correspond to the same areas of activity that I saw. Only I can say they didn't use all those brain areas all at once. They use the mesopallium to learn it, and then they use the tegmentum and the other areas for motor control to perfect it. So those are the 3 studies that I did in order to attempt to earn a dissertation. I thank you very much for coming to my dissertation defense, and now I'm going to open up the floor to you to ask, are there any questions? Thank you.
Nice job.
You're muted, John.
Yep, great talk, Loma.
Thank you. Any questions about any of this?
I have a question.
Yeah.
I'm curious why in the first chapter, which was also about acquiring food, you said more of the males were the models, and then they had the bigger brains and they were able to solve this faster. But then later you said, well, the females have to figure out the tool use to get the food. Why wouldn't they have, why that seems inconsistent to me.
Yeah, I believe that the males were better in the first task because they were the models. They were brought in while they were still hungry, and malnourished is the wrong word, but they weren't as well fed as they were after they'd spent 2 weeks just being fed all they wanted to eat in captivity. So they had the strong motivation to solve it. And they didn't necessarily solve it using the more cognitively advanced strategies. Whenever they were first gained access to it, they used attaching string and food intercept, which is cognitively easy to do, even though it might require some agility or physical strength to get it. Most of the females, by pure chance, ended up in the observer group. They just weren't motivated to get that food because they were already well fed. That's what I think is happening. For the ASOP study, I made sure to restrict their food access more so that they were more motivated to try to solve that.
Did you ever try food restricting the long-term birds?
The string pull task was given to them for the first month or so of captivity. So for the initial rounds of for the initial cohorts, I didn't restrict their foods, but then I learned for the later ones, I do need to restrict their foods, otherwise they're not even going to bother trying to solve this. So I did omit the well-fed group from some of the analyses. I didn't really have time to go over it here, but I did talk about that in greater detail in my dissertation.
And if you compared the well-fed to the food restricted, do you see?
There was not a significant difference, but there was a slight difference, but it just wasn't statistically significant.
So the food restricteds didn't learn faster or sort of stick with it more. As you know, there's a large motivational component here, and that's always tricky to pull out of studies like this.
Yeah, so the main finding there was that the models were better than the observers. When you remove the cohort that wasn't as food restricted, you lessen that effect.
Mm-hmm.
Or I'm sorry, you increase that effect. Shoot, now I can't remember. It's in my dissertation.
Yeah.
I have a question. In the second study, you said you hypothesized that you would see the hypothalamus, but you
Yeah,
didn't.
I did not see that.
So that may or may not be real. And if it is, so can you tell me why, if there might be another, it may have been activated, but you may not have seen it on the imaging.
Right, and that's because there's only so much FDG in the body. And so whichever part of the brain is most active is going to, I guess, hog that FDG uptake, and it will mask activity in other areas. So we're looking at the areas that are most active in response to the rest of the brain. We're comparing it to other scans. And if that same, or let's use two scans as an example. Let's say that the hypothalamus was equally active for both scans. It might have even been more active in the second scan. But because this other region over here in the second scan was even more active, it's drawing the FDG away and it's concentrating it there, and I wouldn't see it over here, even though it was slightly more active.
There's another reason too. It has to do with how we analyze the scans.
Oh, right, we're only looking one way.
Well, and also that the hypothalamus is a small structure.
Oh, right. So if there's a large structure and that entire large structure is absorbing FDG, then it's going to show up more. Whereas this small structure over here, we might dismiss it as an artifact.
Well, and then we have a bias in the imaging methods because sometimes there are slight mismatches, right? So
Right.
if you're off by a pixel and your brain isn't matched up, then a small structure is less likely to show the group-wise activation, compared to the second or the other group, right? Because there'll be a small mismatch in it, and the larger structures always tend to statistically show up a little bit better because the mismatches don't matter as much. Yeah, it's kind of an important point with all this stuff.
Thanks, Donna.
I got a couple questions, Loma. You've shown us the video of the crow solving the ASOP task before, but I'm always struck at just how far down the animal is able to dive to stick its beak in. So I wondered whether you either did, or could you speculate on how best to proceed in further experiments if you or somebody else were going to do it to make the task harder? Would you add, like, vary the number of rocks required by varying the size of the rocks? Would you keep the size of the rocks constant and make the tubes a little bit narrower so that the bird can't dive as much but needs to put in, say, 5 or 6 rocks to get the food item? Which path would you take to sort of push these crows a little bit further in their cognitive ability and see what they're capable of?
Well, I've actually got two answers to that question. One of which is things I did try and dismissed or kept, and things that other people have tried. What I've tried doing is I've added blocks underneath the tube to raise it closer to the ceiling so they can't really fit their whole body up there and upend the way they did. I also ran into issues where, the bird's maximum reachable distance at first changed because they learned that they could dive deeper like that. So I had to readjust and try again. So the solution that I did was I just made it deeper and raise it up to the ceiling. But another thing I could do would be changing the properties of the rocks and the tube. So I can make it more difficult by using smaller stones, or even stones that float and they have to choose which stones to use. I could change the properties of the tube. So I can make it narrow, so they have to use the smaller stones. Or I can make it so that they have to drop it into another one and have a U-bend underneath. So they can't drop it in here, but that's where the food is. So they have to drop it over here and it raises up this way. I could even use a material that isn't displaced by stones, like sand. And they have to choose. I've only got 3 rocks. Do I put it in this tube or this tube?
So you've thought about lots of possibilities. Which would be the way, if you were sort of taking on this task next, what would you do next?
I think the thing that I would do next is one, refine the technique from the get-go. So I use what I described here. Like I had to change this multiple times so the crows just were not learning it. But I think I would probably change the quality of the rocks. So I'd offer them multiple options. So they had to pick and choose the big rocks versus the little rocks. And I would consider that as the first step to seeing how well they understand the properties of this cause and effect.
Mm-hmm.
The sex difference in performance is really interesting. You said it's also been seen in cetaceans and some other primates. Did you say?
Bonobos and chimps.
Bonobo. So, the brains versus brawn is sort of, that's a hypothesis, right? Has anyone ever actually tested this? I mean, is it the socialization or competition effect, or is there some more physiological explanation for the sex difference? I mean, does anyone know? I mean, is there?
To my knowledge, I don't know that anyone's tested this. Yeah.
I mean, that's kind of an important, I mean, it's great observation and very interesting, but seems like an important point.
It is speculation on my end.
I mean, it's surprising, that nobody has dug into this to try to understand what's the base. Are we talking about some behavioral socioeconomic difference here, or is this something inherent to the brain?
Well, I'm looking for a job as a tenured faculty. That could be my in.
Yes, that fundable question, and yeah, I mean, that is intriguing. Well, so how would you go about testing whether, are we in the exam yet, or are we just
Well, I don't know. We still have an audience. I guess we could.
Matt, if you did get a job and you were going to follow, I mean, how would you start approaching that? How would you try to distinguish an environmental effect versus a physiological effect that gave rise to a sex difference in performance?
Well, I think I'd have to do a lot of foundational studies first. Make sure that the species I'm studying could be American crows, it could be a different animal depending on where I go. See whether that animal does in fact have a sex difference in tool use, and is that sex difference present in other cognitive tasks or challenges. Then I would also look into their environment. Are there sex differences in how these animals acquire food or deal with like agonistic encounters or anything like that? I would look for some sort of sex difference that might point to the brain versus brawn question. Maybe the males will fight if there's an intruder in their territory, whereas the females call for reinforcements or allies. Something like that.
Yeah. Well, as a general approach, if you wanted to do more than just observe and try to control environment, I mean, what might you do as a first step for a general approach to teasing out the environmental influences?
I would like to bring the animals into the lab to remove as many potential compounds as possible. But then once they're in the lab, I would try to just mimic any sort of observed differences I saw in the wild. So if I, for example, saw that males used a sort of brute force approach to get food and females used a different approach, would they still do that in the lab setting, and under what conditions would they continue to use that strategy?
And would you necessarily always want to just work with adults that you brought in to the lab? Or?
Oh no, I'd also want to use lab raised individuals. So I know, okay, I know the conditions under which they were raised, so to speak. Do they also show this? Is it a learned or an innate thing?
Right. That would be the first thing I would do.
Okay.
Yeah. So there's a few years of work.
That's true. So I'll start my career.
Yeah. I think you could even change the dominance structure of the groups if you had them in all female or all male groups. and accentuate that or reduce it.
See if the dominant females start to use more brute force approaches, whereas the subordinate females use brain approaches.
Exactly. Yeah.
Yeah.
That would be cool.
Yeah, there's a lot you can do with that.
All right, so it looks like we're going to want to start the exam in a few minutes. Before we stop, are there any other questions from the non-committee members in the audience? All right, well, thank you everyone for coming. And I guess the exam proper will start, but that's going to be us. So thank you very much.